The Affymetrix gene chip is an oligonucleotide array consisting of several perfect match, or PM, and their corresponding mismatch, or MM, probes that interrogate for a single gene. The perfect match probe is the exact complementary sequence of the target genetic sequence of interest and is composed of 25 base pairs. The corresponding mismatch probe has the same sequence as its paired perfect match probe, with the exception that the middle base, in the 13th position, has been reversed. The idea behind the mismatch probe is to assess nonspecific hybridization. There are roughly 11 to 20 perfect match and mismatch probe pairs that interrogate for a single gene, called a probe set. The perfect match and mismatch intensities are combined to form an expression measure for the probe set or gene. We will discuss methods for combining perfect match and mismatch intensities later in Module 6. Affymetrix uses photolithography to light direct bases to the surface of the chip, where a cover is placed to mask or protect the cells that should not get that base. Probes are built up base by base until the probe length is 25 bases. Affymetrix quickly dominated the market back in the early 2000s. Initially, the perfect match and mismatch probes in a probe set were all tiled next to one another on the surface of the gene chip. Problematically, if any artifacts affected the surface of the chip, such as a bubble or scratch, that would usually correspond to the inability to get an expression value for the gene corresponding to that probe set. Subsequently, gene chips were designed such that probe pairs within a probe set were randomly scattered about the surface of the chip. For an Affymetrix gene chip, only the experimental sample is hybridized. There is no need for a control sample. Before we discuss RNA sequencing, first watch this short YouTube video describing DNA sequencing.